Rating questions once he has completed his lecture. Thank you.
Thank you, Dr. Rodriguez, for a very kind introduction. And thank you to everybody for attending today. Like Dr. Rodriguez mentioned, I do have a variety of active research interests in areas of research, but what I wanted to focus on in my talk today is the work that my team has been doing regarding military stress, diet, and the gut microbiome. So I thought I'd just start by giving a little bit of background about who I work for, what we do. As was mentioned, I work for the Army Research Institute of Environmental Medicine, or USAREUM, and specifically within the Military Nutrition Division, where we have a research mission of conducting research that provides a biomedical science basis for developing new rations, menus, and nutrition policies and programs that enable warfighter health, readiness, and optimal performance in any environment. So there's a few key things there. We are not the people who actually develop military rations. We are the science behind the rations. So we do the science that gets into the recommendations that dictate what warfighters get fed. And then we focus primarily on performance, not so much chronic health, but day-to-day performance of our military personnel. And we are located in Natick, Massachusetts, on this wonderful campus here that kind of is reminiscent of the state of Texas. And we are actually a relatively small institute, maybe 300 people in total, that occupy this single building on the campus of the Natick Soldier Systems Center. All right, so why would a military nutrition division have interest in the microbiome and the gut microbiome in particular? Well, it's no secret that the gut microbiome and interest in the gut microbiome has exploded in the past 10 to 20 years. I always, when I'm in person, I always like to ask the audience, you know, especially the students, who has heard of the microbiome? And nowadays, everybody will raise their hand. When I was a student back at, especially getting my bachelor's, I had no idea what the microbiome is, never heard of it, but it's everywhere now because of high impact scientific publications, publications in popular press. You see it on TV. You have books about the microbiome now. You have citizen science projects about getting your microbiome sampled. There's now companies that are reporting to be able to analyze your microbiome and make diet-based recommendations. Based on that, I won't get into my opinions on that in this talk, but they're out there. It's even made it onto South Park, which tells you that microbiome is really mainstream. Now, why is that? Well, it really boils down to the fact that we as humans are actually more microbial from at a cellular level than we are humans. So if you were to add up all the cells on and within the human body, you would find that actually there's more microbial cells than there are human cells by a small portion. However, if you look at it from a genomic perspective, what's really astounding is the number of genes in the microbial genome, the human microbiome relative to the human genome. It's orders of magnitude larger, meaning that the microbes living on and in us are capable of carrying out functions that our human bodies cannot. Now, it's true that each individual microbe is just kind of a small individual unit, has a small genome, not capable of doing a ton of work on its own. However, when we think about microbiomes and microbiotas, it's important to recognize that these microbes live in diverse, dynamic, multifunctional, and complementary communities. They're working together, sharing resources and functions to coexist in the human body and on the human body. And these microbial communities are connected throughout the human body. And I like to think of the gut microbiome as kind of the central hub of all the microbiomes on the human body. It's by far the most dense collection of microorganisms on the human body. So it's kind of like a global communications network, making signals, constructing signals, and sending them out not only to other microbiomes in the body, but also organ systems in the human body. So the gut microbiome in particular modulates the immune system, interacts with the enteric nervous system and the vagus nerve, and produces myriad metabolites that act not just in the gut, but enter circulation to affect organ systems all over the human body. So in this way, the gut microbiome is communicating not just with other microbiomes, but with organ systems throughout the body. And what that means is that these microbes have the capability of potentially modulating functions of those organ systems, affecting things like mood, behavior, cardiovascular function, how we metabolize and use energy, how our intestinal barriers functions, and even things like muscle repair and bone health. And if you're not convinced that microbes can really have an impact on the human body, just think about food poisoning as an example. So food poisoning or traveler's diarrhea are induced by single strains of microbes that are able to gain a competitive advantage in the gastrointestinal environment. So that's a single microbe having effects on the entire body. So if you don't think the gut microbiome matters, just think about the power of those specific pathogens that can influence how we feel all over our entire body. The question is, can we modulate our commensal gut microbes, the ones that live within us on a day-to-day basis, in a way that benefits health? And the other important thing about the gut microbiome is that it's malleable. And this is exciting. I'll tell you why it's exciting from a nutrition perspective. It's because we know that diet is one of those things that modulates the gut microbiome. But it's also true that other things that we're exposed to in our day-to-day lives, what we like to call the exposome, with we like to put "ome" on everything these days. So exposome is another example of that. But from a microbiome perspective, the things that we're exposed to in our environments, how physically active we are, how much stress we have when we're preparing for exams or trying to submit a grant proposal, medications that we may take, antibiotics to deal with infections in particular, our sleep cycles, not getting enough sleep, circadian disruption, and environments all modulate the microbiome and can put it out of whack or control it, put it in a state that's more beneficial to health. So we have these environmental factors interacting with a system that is impacting various organ systems throughout the body. All right, so what I want to cover today with that introduction is why we care about the microbiome from a military nutrition perspective. I alluded to it a little bit during those previous slides, but I'll get into more details over the next few slides. And what I really want to focus on today is some of the evidence that our group is starting to develop, showing that the microbiome probably is relevant to warfighter health and performance, and then I'll talk about finally what we intend to do about that, and how we intend to use nutrition in particular to leverage this tremendous functional potential of the gut microbiome in a way that benefits the health and performance of our military men and women. All right, so this word, the exposome, again. What is the exposome from a military context? Well, in the work that we do, the military exposome includes combinations of stressors that any one of us may experience to some degree, maybe in isolation. However, warfighters and military personnel tend to experience these stressors in combinations and at extremes, which makes them a fairly unique population. And those stressors include prolonged work, so being active over very long periods of time, mostly kind of moderate intensity activity interspersed with some high intensity work. There's a very big component of psychological stress depending on the environment. I'm sure you can all imagine some of the psychological stressors that military personnel are exposed to. There's sleep deprivation, circadian disruption, environmental extremes like high altitude, heat and cold, austere environments where they're living essentially off the land at times. And then undernutrition is something that we think about a lot. During training and combat, military personnel often do not eat enough, lose muscle mass and body mass as well, which can have negative health and performance consequences. So when we started this work, when I started this work, and maybe 5 or 6 years ago now, there was a lot of provocative evidence, primarily from animal studies, indicating that those exposures that I just talked about can interact with the microbiome in a way that impacts host health and performance. However, the bulk of that evidence was derived from animal models, and translation of those findings to humans was largely lacking, at least in my opinion. So one of the things that our program aimed to do is to first understand how the military exposome impacts the gut microbiome and what that might mean to health and performance of military personnel. And the second part of this is from a nutrition perspective. We know that what we eat shapes the gut microbiome. And it's not just about the nutrients in the diet, it's also about how our foods are prepared, how well cooked they are they raw, are they heated, How processed are they? What additives are added? How ripe the food is? All of these characteristics and nuances about what we eat in our diet influences the nutrients that get to the large intestine where the gut microbiota resides. And it's not just about what we eat, it's about how we eat as well, how well we chew our food, how long it takes food to get through the small intestine and large intestine, our individual differences in our physiology and digestion. Ultimately, all these factors come together to impact the nutrients that reach microbes in our gut. And if you think about back to one of the first slides I introduced, the genomic potential of the gut microbiome, and the fact that these microbes can carry out functions that our human bodies cannot, a lot of those functions involve interacting with the nutrients that we don't digest. They convert these nutrients into various metabolites that have beneficial health effects, like short-chain fatty acids, other metabolites that have neutral or to be determined health effects, secondary bile acids, for example, some metabolites of protein fermentation, and then myriad other bioactive compounds that are largely beneficial to health, but also some of which seem to be detrimental to health. So when we put it all together from a military nutrition perspective, we have the gut microbiome, which is trillions of active microbes inhabiting the GI tract that influence organ systems throughout the body. We know that operational stressors and diet that our military personnel are exposed to impacts the microbiome, and we know that the microbiome influences how the human body responds to diet, environment, and stress, which makes the microbiome and how it interacts with the military exposome relevant to our pursuit of optimizing warfighter health and performance. And some of the outcomes we may be interested in, some of the outcomes that we think about as a research group are related to the microbiome and this interaction with the exposome. Things like psychological health and cognition, respiratory tract infections, traveler's diarrhea, and what we'll talk about a lot today, gastrointestinal permeability, maybe even stress fracture and physical performance. So these are issues that we think about from a military nutrition perspective that seem to be potentially linked in some ways to the microbiome and potentially modifiable by leveraging the microbiome through nutrition to ultimately impact health and performance of our warfighters. And that's really the second knowledge gap my research program intends to address. One, can we establish a link between the exposome and the microbiome and warfighter health and performance? And then two, how do we use nutrition to leverage the power of the microbiome in a way that optimizes health and performance? How do we go about doing this? Well, we do this through both field studies and laboratory studies. So really our bread and butter, I like to say, is going out to the field environments, working with warfighters in the environments in which they train and operate to really understand what's going on in those austere environments. But we also do clinical studies to help us interpret the findings we see out in the field and develop mechanistic insight and generate hypotheses that we can then go out and test in field environments. So from the microbiome perspective, we tend to focus on a few things, and I'll be sharing results from studies that we've conducted that deal with each of the components that I'm going to share with you now. So what we rely on a lot, you can't be in the microbiome field without having to be comfortable handling fecal samples. So we do that because that's the only way we can really non-invasively study in humans the gut microbiota. Most of our research focuses on the technique called 16S ribosomal RNA gene sequencing, and this is a technique that helps us understand the types of microbes present in the gut microbiota. I won't show you any data today, but we are starting to get into shotgun metagenomics, which is sequencing the entire genome of the microbiota to understand the microbiome. So not only who is there, but the functional potential of that community. We also do a lot of work with both targeted and untargeted assessments of metabolites that are found in fecal samples, many of which are modulated or only produced by the gut microbiota. Some of the targeted metabolites we focus on are short-chain fatty acids in particular. These are compounds produced by gut microbes, primarily through carbohydrate fermentation, but also amino acid fermentation as well. And they're largely beneficial for host health, or at least thought to be beneficial for host health. So we really focus on these as potential modulators of the gut microbiota human physiology interaction. We also tend to measure it, well, we also measure intestinal permeability and really all of the microbiome studies that we've done thus far. And I'll be sharing you results of those analyses in the presentation today. And we focus on intestinal permeability because the intestinal barrier is really at the interface between the microbial world and the intestinal lumen and host physiology. And a breakdown in that barrier is important because, as you can see in this diagram here, it can allow bacteria or bacterial products to get into circulation, where they stimulate immune responses, stimulate inflammation in a way that can have negative effects in organ systems throughout the body, to include potentially in the brain, impacting cognition and behavior. And the way we measure intestinal permeability in these studies is by trying to do it directly using what we call a dual sugar absorption test, where we feed concentrated doses of multiple artificial sweeteners and look at ratios of the sweeteners as they come out in the urine over a period of 24 hours. And that gives us an indication of how permeable the intestinal barrier is. We also measure blood and focus on biomarkers in the blood that are indicators of host physiology and how that physiology changes, and try to link those biomarkers back to some of the features we see in the microbiome. So biomarkers of inflammation, gut barrier function, and gut barrier damage are commonly measured, and I'll show you some results of those analyses today. And then we also focus on both subjective and objective responses in these environments in an attempt to try to link changes we see in the microbiome and changes we see in gut barrier function to objective and subjective ways of measuring performance. So things like, how do you feel, and how are you physically performing? I'll focus primarily today on some of the subjective responses in relation to the microbiome gut axis. And we think about the microbiome in this context from really two frameworks. One is kind of a mediation model, where you have our exposures changing the gut microbiome in a way, and that change of the microbiome induces it in a biological response that in turn impacts health and performance. But we also think of it from an effect modification type of approach, where our native microbes are influencing our biological response to an exposure. So it's not the exposure impacting the microbiome, it's the microbiome influencing our physiologic response to that environment, thus impacting health and performance. And I'll come back to this kind of concept at the end of discussing each study that I'm going to share with you today. All right, so let's talk about the first study that was really our first major foray into trying to understand knowledge gap 1 that I talked about earlier, the interaction between military stress and the microbiome, and what impact it might have on warfighter health and performance. So this was a study that we conducted in collaboration with the Norwegian Defense Forces back about 5 years ago now. We are gearing up to go out there again to try to repeat some of these findings, and we're all very excited about that in the next week or so. But we're going back in part, at least I'm going back in part, because of the findings I'm going to share with you today, which were really intriguing and helped establish the line of research I'm sharing with you now. So the environment we were studying is what I would call a multi-stressor environment, which is not atypical for military personnel. If we think back to the exposome, this is characteristic of the military exposome, where you have multiple stressors experienced in combination at the same time. In this study, it was a cross-country ski march, so it's about a 5-day ski march or a 4-day ski march over 50 kilometers. So we have a low, moderate intensity exercise. You can see the heart rates plotted on the graph in the middle here, where they're not too high, but they're fairly sustained for a long period of time over the day. They're out in this Arctic environment in the middle of winter. They're not sleeping very well. They have a shift in diet from their normal diet to military rations, and they have a psychological stress of needing to complete this course, and also they're being evaluated on how well they perform. And an unexpected source of psychological stress in this environment was a blizzard that came up during the middle of the training that stranded some of the soldiers on the mountain. So all sorts of things going on that can induce stress. And the other thing is this undernutrition that we see a lot in field environments. So they were burning, and this was measured using doubly labeled water, 6,000 calories per day energy. So that's a huge energy expenditure, and they're eating 3,000 calories per day, which seems like a lot, but when you put it in perspective of the energy being burned, they're in an energy deficit, which causes body weight and muscle mass loss. I will mention that there were 3 different diet groups in this study. The primary outcome was focused on a diet intervention, but it had no effect on the outcomes I'm sharing with you today. which relate to microbiome, intestinal permeability, and blood markers measured at the beginning and end of the training. So what did we see? Well, first we saw a pronounced shift in gut microbiota composition. And if any of you aren't familiar with these kind of plots, they're relatively simple to understand, but take a little bit of time to figure out what's going on. Each of these points represents the entire microbiota community of one individual at one point in time. So the circles are samples taken before the ski march, diamonds are samples taken after the ski march. Samples closer together are more similar than samples farther apart. So what we see here is all the pre-samples are kind of grouped together on the right side of the graph, and all the diamond-shaped samples are grouped together on the left side of the graph. What that's indicating is a huge shift in microbiota composition. And the shift that we're seeing in this study is actually pretty astounding. You don't see this kind of discrimination often in human studies. Maybe you do in animal studies from time to time. But normally human studies, samples from the same person tend to group together and obscure some of the effects of the intervention or the exposure. Not at all what we saw in this study. There's really dynamic shifts in microbiota composition, to include some reductions in what we consider beneficial bacteria, such as these notated here, Faecalibacterium or Roseburia. These are short-chain fatty acid producing microbes. They produce the short-chain fatty acid butyrate, which is well known to have beneficial health effects, particularly in the gut. All right, so we have a change in microbiota composition. We also have a change in both the fecal and plasma metabolome that was related to the change in microbiota composition. So these graphs here in the middle are just graphs that are basically looking at a correlation between the gut microbiota and the metabolome being all the metabolites, and there's hundreds of them that we measured in the fecal samples and the plasma samples. Those middle two graphs are just showing that there's a relationship between the two. The microbiota is related to both the stool metabolome and the plasma metabolome. And what we were able to do is link some of those metabolites to changes in microbiota composition. In particular, there were 69 metabolites that changed in response to the training that we were able to show to be linked to changes in microbiota composition. So not only is who is present in the microbiota changing and their relative abundances with respect to each other, but the metabolites that they seem to be produced are also changing both in the gastrointestinal tract, but also in the bloodstream as well. The other thing we saw in this study was an increase in intestinal permeability. That's plotted here from pre to post, about a 55 to 60% increase in intestinal permeability. And that was important because we were able to link that increase in permeability to an increase in inflammation. And inflammation is something we try to control in a military training environment because it could have potentially negative consequences. And we see a relationship between this increase in intestinal permeability and increases in inflammation measured by IL-6, blood IL-6 concentrations in this case. Now, I can't say that the permeability increase caused the increase of inflammation, or whether or not the increase in inflammation preceded the increase in permeability in this case. But nonetheless, there is a connection here between inflammation and permeability. Just the directionality is a little unclear from the study design. All right, so is the microbiota involved in this change in permeability and inflammation? Well, it turns out we were able to link changes in microbiota composition and metabolite production, and also the pre-training microbiota to this increase in intestinal permeability. So if we think back, I told you we'd like to think about the microbiota from both a mediator and effect modifier perspective. So in this study, from the effect modifier perspective, we saw that people's microbiota composition before the training environment was associated with the increase in intestinal permeability during the training environment. So soldiers who had a more diverse gut microbiota actually had a smaller increase in permeability in the military training scenario. And this is consistent with the general concept, not an exclusive concept, but a general concept that a more diverse microbiota is a healthier and more resilient microbiota. And that's kind of what we're seeing in this case. It's offering protection. Conversely, we see a higher relative abundance of this proteobacteria, which is a phyla, which is a relatively general characterization of a group of bacteria. We see a higher proportion of those bacteria are associated with larger increases in permeability during training. And that's consistent with the fact that many bacteria, if not all bacteria in that phyla, are known to produce lipopolysaccharide. Lipopolysaccharide is a pro-inflammatory stimulus. You don't want that getting into your bloodstream. It's also associated with inflammatory GI disorders. So that association is consistent, has some biological plausibility behind it. Then we also saw decreases in several metabolites that are potentially linked to gut barrier function being associated with larger increases in intestinal permeability. So the gut microbiota can produce essential amino acids and other amino acids, and we were able to link amino acid concentrations in the fecal material to gut microbiota composition, and saw decreases in two that are involved in antioxidant effects or barrier function were associated with increases in permeability. And then we also had we observed something that I'll come back to later, and that decreases in two secondary bile acids in particular, and secondary bile acids are produced by gut microbes and their actions on primary bile acids. Reductions in these two bile acids were associated with increases in intestinal permeability. And it's thought that modulation of the bile acid pool, in particular ratios of primary, so that's what the human body produces, and secondary bile acids, the bile acids the microbial community produces, impacts gut barrier function, and that we're seeing some signs of that here. So if we put that all together, we have an Arctic military training environment altering the microbiota, including reducing relative abundance of some of these beneficial microbes, altering metabolites that are linked to the microbiota in a way that is associated with increases in intestinal permeability and inflammation. We also see evidence of effect modification where the pre-training microbiota, how diverse it is, and the relative abundance of these pro-inflammatory taxa are also associated with the physiologic response of the environment, that being increases in intestinal permeability and inflammation. So we followed that study up with another study conducted in an austere environment, and this was a study conducted at high altitude. So we have the physiologic stress of high altitude, and if any of you have ever been to high altitude, I think you'll understand what I'm talking about. But when you go to high altitude, it's really a severe physiologic stress on the body. The reduced oxygen availability to tissues in your body results in sickness in a lot of people. 80% of people, in fact, at the altitude we were at, which was 4,300 meters or 14,000 feet. The other stressor in this study was a severe energy restriction. When people go to altitude, they almost inevitably undereat because you lose your appetite. We designed the study that way to underfeed individuals, to study the interaction between altitude and underfeeding on some of the study outcomes. And you can see in this graph here in the middle of the screen, our participants lost 8 kilos over 3 weeks. So it was a fairly severe energy deficit that they were exposed to. There was also a dietary component to this study. And I should tell you that these studies, microbiota analyses were secondary outcomes. They were not the primary outcomes in the study. I was fortunate enough to be able to include these outcomes, and I'll show you how that's kind of led to our research program building upon these observations. But in this study, the primary outcome was focused on this intervention of standard and high protein diets in mitigating muscle mass loss at high altitude when underfed. Again, in this case, the high protein diet compared to the standard protein diet didn't have a substantial impact on the data that I'm sharing with you, so I won't talk a whole lot about that. What I will talk about are the fecal, blood, and urine samples collected both at sea level and progressively throughout the 3 weeks of high altitude exposure that were included as part of this study. So again, we have another one of these plots where each of these data points represents the entire microbiota community of one individual at one point in time. The HP abbreviations are high protein diet, SP are standard protein. Again, that had no effect on the outcomes I'm sharing with you today. SL stands for sea level, high altitude stands for, HA stands for high altitude. And it's a little hard, it's much harder to see on this graph, but this is more characteristics of what you often see in human microbiota studies, is that there's no real obvious pattern in clustering of samples according to time points. So the lighter shading being the earlier sea level time points, the darker shading, the darker reds and blues being the altitude time points. You do see a little bit of clustering over here in the lower right-hand corner, and there was actually an effect of time over the study where the microbiota became more similar among individuals over time, but not really an obvious effect like we saw in the last study. We do also see evidence of a decrease in beneficial bacterial taxa. In this graph, we're looking at lactobacillus, which is a beneficial microbe. You often see it used in probiotics. It has a number of health benefits different species of lactobacillus do. And we saw a reduction in these species during the high altitude exposure, particularly after high altitude 2 was out after 1 week of exposure or 10 days of exposure, high altitude 3 after 21 days of exposure. And in this study, we didn't do an untargeted metabolomics analysis. We did a more focused targeted analysis focused on fecal short chain fatty acid concentrations. Again, these are the beneficial metabolites produced by gut microbes. And we saw generally a slight decrease in these short chain fatty acid concentrations at high altitude, which is probably to do more with the underfeeding and not having as much substrate to metabolize than the high altitude environment in this case. But we do see a reduction in short chain fatty acid concentrations. We also saw an increase in intestinal permeability, again, about a 55 to 60% increase in intestinal permeability. That was observed within 24 hours of exposure to this high altitude environment, so even before they started on the diet part of the study. And then it was sustained over 18 days at high altitude. So there does, and this is consistent with some animal studies that have shown that high altitude exposure can really have a profound and detrimental impact on gut barrier function. We're seeing signs of that here and over the course of the study. And we were able to show an association between that increase and that decrease in short chain fatty acid concentrations. So if we look at fecal acetate, we see that more fecal acetate is associated with smaller increases in intestinal permeability. Again, supporting the idea that these short chain fatty acids are beneficial and potentially have a beneficial effect on gut barrier function. And when thinking about how the microbiota may impact the physiologic response at high altitude, we went back to this effect modification model to see if the microbiota may influence how people respond subjectively to the high altitude environment. When we measure subjective responses to altitude exposure using a questionnaire that measures what we call acute mountain sickness. So acute mountain sickness is a constellation of symptoms: nausea, vomiting, headache, loss of appetite, that range in severity from really none to very severe, severe enough that it can be life-threatening in certain situations. What's interesting about acute mountain sickness is there's a tremendous amount of inter-individual variability, meaning that people respond differently. Some people get very sick, other people don't get sick at all at the same altitude, and we really don't understand why. We were interested in whether or not the microbiota may play a role. So if you look at this graph here on the left, what we did is we divided people into, based on their questionnaire responses, those who are experiencing moderate or severe AMS, or no versus mild AMS, and asked, did their microbiota composition differ before they went into altitude? And indeed, we found some interesting results. So in this slide, I'm showing that Prevotella relative abundance was higher in people who had more severe AMS. And this is interesting because Prevotella is a genus that we know shows substantial inter-individual variability across human populations. Some people have a ton of this genus, and other people have none. So it's really a gradient between none and very high amounts across human populations. And that genus has been associated with diet composition, and has also been associated with inflammation, glucose homeostasis, and a variety of other health outcomes. So what we're seeing here is that this inter-individual variability we see in gut microbiota composition across people may actually be associated with how these individuals are responding to a high altitude environment. And we're doing some work now to try to reproduce that finding in larger populations and different populations. So again, in this study, we see evidence of mediation, the energy deficit at high altitude being associated with a decrease in lactobacillus, fecal short chain fatty acid concentrations, and increases in intestinal permeability. And we see this effect modification of Prevotella perhaps influencing the severity of acute mountain sickness at high altitude. So those two studies really provided us a strong rationale for continuing this line of research into whether the microbiota matters, and if so, how we can leverage the microbiota for warfighter health and performance. Those were both, however, field studies that are observational in nature. So while they're useful for generating hypotheses, we need to interpret them with the appropriate caution, recognizing that these were secondary outcomes in an observational setting, and we're just looking at correlations. So what we decided to do next is really try to understand the factors that may be driving some of those changes in microbiota composition, in the fecal metabolome, and changes in intestinal permeability that we saw in those two studies from a more controlled standpoint. And the first thing we decided to focus on, being a nutrition group, is what we feed military personnel. And what we feed military personnel in training and combat operations is primarily what we call the meal ready to eat ration. And you can see pictures of that here. It's a highly processed food product. There's 24 different menus, each contain an entree, some side items, snack items, candy, desserts, not a whole lot of fruit, not a lot of fermented fruits, not a lot of vegetables, and a very highly processed diet. We decided to isolate the effects of that diet on the gut microbiota and the metabolome and intestinal permeability by conducting a controlled feeding study at our laboratory in Usarium. We enrolled 60 individuals, assigned 30 of them to follow their usual diet and did diet records to understand what they were eating. And we assigned the other 30 to eat nothing but this meal ready to eat ration for 21 days. So these individuals were provided food at a level, the meal ready to eat ration at a level intended to maintain their body weight for 21 days. And then we collected fecal, urine, and blood samples throughout the intervention at the time point shown on the screen. And if you're curious about what the macronutrient content of an MRE diet looks like, you can see that on the slide here. Based on the methods that we had available to measure dietary intake in this study, the MRE participants, while they're eating the meal ready to eat diet, were eating in absolute amounts more protein, more carbohydrate, less protein, more fat, saturated fat, and polyunsaturated fat than people eating their normal diets. And what surprises a lot of people, but probably shouldn't surprise us, is that people eating the MRE ration were actually eating more fiber than participants eating their usual diet, which is generally consistent with a really poor practice of Americans in not eating near enough dietary fiber, which is an important nutrient for maintaining a healthy gut microbiota. All right, so in this study, we did see that the MRE diet had an effect on gut microbiota composition. The genera affected are shown here, and I'll just make point out a couple of interesting observations. These bars in yellow are genera that were decreased in the MRE group compared to the control group. So that's Lactococcus, Lactobacillus, and Leuconostoc. All of those genera are used in fermented food products, cheese, yogurt, things that are not present in the MRE diet. Lactobacillus in particular is a healthy microbe that you find in fermented foods. So it's really no surprise that you're seeing reductions in abundance. So these are lower in abundance in individuals eating the MRE diet. So we're seeing that, probably habitual diets and the amount of fermented foods people ate impact the response to an MRE diet because those same foods are not present in the diet. We also see some other microbes that were increased in the MRE group compared to the control group. Clostridium is a very diverse genus involved in bile acid metabolism, also protein fermentation. And then Veillonella is an interesting one that metabolizes lactate and produces short chain fatty acids, which were enhanced in the MRE group. We did not see an effect on fecal short chain fatty acid concentrations, which may not be surprising given the fiber results I shared with you and the fact that the MRE participants were actually eating more fiber, which is the primary source of fecal short chain fatty acids. But we did see, when we did untargeted analyses, we did see changes in the fecal metabolome. So the short chain fatty acids being a targeted analysis, the fecal metabolome being a measurement of all the metabolites that we can in the fecal material. So what I'm showing you here is new data that I'm really excited to be sharing with you because it hasn't been published yet. And it's really sparking some excitement in my group at some of the findings that are coming out with this. So what we're looking at this plot, so we're not looking at microbiota data here, the plot on the right. What we're looking at is metabolomics data. So each data point here is the entire metabolome of one individual at one point in time. Or this is the change in the metabolism from metabolome from pre to post intervention from one individual during the intervention period. The open circles are the MRE group, dark circles are the control group. Again, samples closer together are more similar than samples farther apart. You see this clustering of MRE participants over here, control participants over here, telling us that the changes in the fecal metabolome for pre to post intervention differ between the groups. And we are again able to use this analysis shown in the graph on the left to show that that change in the fecal metabolome was indeed associated with gut microbiota composition. So we're seeing another interaction between diet, the microbiome, and the fecal metabolome. So what does that matter? Well, to start, let's look at the measures of gastrointestinal barrier function and inflammation that we measured in this study. So if you recall the previous two studies, we saw increases in intestinal barrier permeability in austere environments. In this study, when we focused just on the diet, and this is shown in the panel on the left, we actually saw a decrease in intestinal permeability in the MRE group compared to the control group, which tells me that the increase in permeability we see in the austere environments is not because of the diet that we're feeding our military service members. It's probably because of the physical stress of the environment itself, which we know can damage intestinal barrier function. If you look at panel D down here in the lower right, we're using C-reactive protein to measure inflammation in this sample. And we see also that a reduction in inflammation in the MRE group, that's a solid line, compared to the control group, that's a dotted line, which coincides with this decrease in intestinal permeability. So that's a good thing we're seeing as a result of the MRE diet. The other outcomes we're measuring, intestinal fatty acid binding protein is a measure of gut barrier damage. Glucagon-like peptide 2 is a beneficial hormone produced by the GI enterocytes in the gastrointestinal tract that benefit GI barrier function. Lipopolysaccharide binding protein is a compound produced in the liver to bind lipopolysaccharide getting into circulation. We see kind of what I would call a mixed response here. So some of these changes, like the reductions in IF-ABP, are beneficial on the MRE diet. The reduction in GLP-2 maybe not, and no effect on LBP. So what we're seeing here is overall kind of an improvement in intestinal permeability on the MRE diet and a more nuanced, not quite clear effect on some of these other markers of GI barrier function. So what we wanted to know is whether we can understand this variability that we see in these responses. And I can give you an idea of what I'm talking about by focusing on this outcome here. This is irritable bowel syndrome symptom severity score. So this is a measure of overall gastrointestinal discomfort measured by a questionnaire. And what I'm showing you here are changes from baseline to post-intervention in the MRE group. You can see there's a variety of responses where some people here on the left are having decreases in IBS scores, others having increases. And this gray is the change in the average change in the control group. So you can see that some of these people in the MRE group are having a response greater than what you would expect based on normal variation, others having a decrease, inter-individual variability. Can we understand, is that due to differences in microbiota composition? Well, it seems like maybe it is. So the graph I'm showing you here on the left is an analysis that tries to relate the fecal metabolome and changes in the fecal metabolome to changes in all these measures of GI barrier function that we're looking at. And each of these blue lines represents a collection of interrelated or correlated and anti-correlated metabolites measured in fecal samples, the changes in the metabolites. Each of these red lines indicates changes in a marker of GI barrier function, or for example, questionnaire-based responses is IBS symptom severity score. And then each of these individual data points is one individual, one individual. Basically what this analysis is showing, without getting into too much detail is that there were certain metabolites. The changes in these metabolites were correlated with changes in our markers of GI barrier health and function. And we were able to link changes in those metabolites to changes in microbiota composition. And that's shown by the same analysis here, only now we're looking at correlations between changes in microbiota composition and changes in the fecal metabolome in these collection of fecal metabolites. This observation, what I'm showing you here on this slide, is one of the things that I'm excited about now. I won't pretend that I completely have it figured out, but what we're seeing based on the two analyses I showed you is that changes in microbiota composition are related to changes in fecal bile acid profiles. So we have primary and secondary bile acids that were changed by the MRE diet, and those changes were associated with changes in bacteria that we know biotransform and metabolize these bile acids. And those interactions are in turn associated with changes in GI barrier function. So this is consistent with the idea that potentially changing the bile acid pool and the ratio of primary and secondary bile acids by virtue of changing the microbiota may be influencing how one responds from a gastrointestinal perspective to the MRE diet. And that's something we're following up on now, whether or not we can understand how these interactions between changes in the microbiota are modulating bile acid metabolism in a way that's influencing GI barrier function on an MRE diet. All right, so if we revisit our knowledge gaps, we started with how does the military exposome impact the microbiome, and can we use nutrition to target and leverage the microbiome to improve health? So if we go back to the model I showed you before, where we have the microbiota, the intestinal barrier, and being linked to inflammation and potentially brain function, we've seen that strenuous training and high altitude are stressors that can impact that seem to impact intestinal permeability and inflammation. And I just showed you that the MRE diet may actually have some counteraction on that effect, where the MRE diet induces a decrease in permeability and a reduction in inflammation, which is very interesting. But also means that which is a good thing for us, because it means we're not doing any harm with the MRE diet, and potentially we can start tweaking the diet in a way that leverages the microbiome more to prevent even further some of these intestinal permeability changes we see in arduous training environments. And then I showed you that things like short-chain fatty acids, bile acid metabolism, and specific changes in microbiota community composition are associated with these physiologic effects that we see in response to these military training environments. So again, short-chain fatty acids, bile acids, lactic acid bacteria like lactobacillus, diversity, and pro-inflammatory taxa have all emerged as themes that may be relevant when we start thinking about how to target the warfighter microbiome for performance optimization. So where are we going next? Well, we're using those data to try to develop interventions that target some of the signals that we've seen. Targeting lactobacillus, other beneficial microbes, trying to decrease relative abundance of potentially harmful microbes, and augment the production of these beneficial metabolites like short-chain fatty acids. Polyphenols are ones that I haven't mentioned a lot, but which we're very interested in because they are modified by the gut microbiota and have a variety of health benefits. And so our ongoing work, I can't wait to be able to share results of these studies with you, but our ongoing work is focused on using nutrition interventions, primarily using prebiotics. So prebiotics are fermentable substrates, or substrates that are target specific microbes that are beneficial, and that targeting of these beneficial microbes has a proven health benefit. Many prebiotics are fermentable fibers, fructooligosaccharides, inulin, galactooligosaccharides, and then also phytonutrients, polyphenols in particular, which you'll find in plant foods, are also considered prebiotics with beneficial effects on microbiota composition. We're now using an intervention that's enriched in prebiotic compounds to try to modulate the microbiome in a way that prevents the increase in intestinal permeability we see in a high altitude environment. We also have ongoing work using probiotics, which we're very interested in, which are live beneficial microbes shown to have a health benefit to modulate the microbiome in the human in a way that benefits health and performance. So I'll stop there and thank Dr. Rodriguez again for the invitation and all of you for listening. I have to thank my collaborators. This is everything I shared with you is a team effort. I want to give a shout out to two UConn alums in particular, Dr. Pasiakos and Dr. Margolis. Dr. Pasiakos has been a mentor of mine, and most of the research I showed you today, the two studies I began with in particular were his studies that he allowed me to tag on to and really helped us develop our research program. And then the other DoD and university labs that have contributed to this work, including the Army Research Development Command Soldier Center, the Walter Reed Army Institute of Research, Johns Hopkins, Pennington Biomedical Research Center at LSU, and their Norwegian Defense Forces for helping. And I'll leave you with Napoleon's famous quote that an army marches on its gut microbiome. And I think I left a little time for questions. So I'd be happy to take any at this time. Thank you very much.